see the numbers are climbing right now. So we're at 133. We also have a YouTube feed that is live and people can watch us there as well. I will tell you a few words about the TINS Neuroscience Seminar Series. So this is our first edition. This is the kickoff, so be indulgent with us. We don't know how things work yet, so we hope everything will work out fine. This seminar series was born at the initiative of researchers in TINS, which is the Transylvanian Institute of Neuroscience. It is a research institute in Cluj-Napoca, Romania, which is a wonderful city in Transylvania. We here at TINS feel that neuroscience is still poorly represented in many countries across the world, and it deserves more coverage here in Romania, and why not across the world in a free and available manner. We aim to bring neuroscience closer to those who share this passion for it, and in either way, whether they work in this field or not, and we want to inspire more people to become curious in the matters of the brain. Our plan is to hold free online seminars periodically, once or twice every month. And we aim to bring a wide variety of speakers from all corners of neuroscience. The format for today will be a 40-minute talk and followed by a 15-minute or so Q&A section. Please post any questions you might have in the Q&A panel, which should be at the bottom of your screen, or use the chat if you want to have it interactive and make Florian answer immediately. You can, in the Q&A section, you can upvote or comment on existing questions, so that makes it easier for us to manage at the end of the talk for the Q&A section. We will select a couple of interesting questions and Florian will answer them. I think that's it with the organizatorics. So I will now give the word to Florin Albeanu, which is a professor and researcher at Cold Spring Harbor Laboratories in New York, and he will be our moderator for today. So thank you.
Thank you, Adriana. Thank you, Florian, for showing up. It's a pleasure and honor and a privilege to introduce the speaker today, Florian Engert. Florian is one of my scientific heroes, and his presence here, even if only virtual, triggers indeed warm feelings. Florian is quite unique in his ways he goes about in life and in science. In science, I would say he is truly inspirational through his passion, through his incessant curiosity and voracious appetite for discussion and knowledge. And I would say that this brings together a few of the features I would find fundamental in scientific pursuit, that is always questioning the settings, disregarding bullshit, asking simple, clear, and pointed questions, engaging in exchange of ideas with the people around, and always having fun, always enjoying the long pursuit. Florian did his undergrad and his PhD in physics at the LMU in Munich, and while playing with lasers, he got interested in neuroscience, started to ask questions about how the brain works, about the substrates of processes such as learning and memory. Indeed, along these lines, he did a postdoc also in Munich with Tobias Bonhoeffer, and a second one with Muming Pu at UCSD, and then at Berkeley. And then in 2002, I believe, he started his lab in Harvard, where I got to love him very much. Florian's research is focused on behavior, understanding behavior and the neural substrates of behavior, taking a perspective that involves different levels of understanding, from the computation to the algorithms to the actual implementation. And along these lines, actually, his lab is probably the first that has monitor the activity of all the neurons in a given animal, in this case in a fish, a feat that he considers bullshit, but actually has inspired a lot of other pursuits, including some major funding initiative in the States, like the Brain Initiative, which is still ongoing right now. And Florian is himself. So without any further ado, I would say that he has been so engaged in these discussions and teachings that he has never missed a single edition of this summer course we have started 8-9 years ago, TINS in Cluj in Romania. And every time he manages to get lost in the hills of Transylvanian countryside, and every time he manages still to find his way back and hence he's back here ready to give his talk. So, Florian, the stage is all yours. Enjoy.
Well, thank you, Florin. It's extremely flattering introduction. I hope I can measure up, at least in some way, to the extremely high expectations you've set now. Maybe I should also give a little pitch to the TINS Neuroscience Seminars and the workshop that Florin has founded and is organizing every year. If this should ever happen again once this plague is over, then please go check it out and come. It's really awesome. Yeah, I'm gonna share my screen, and can you all see that? Yes. So I'll talk about fish feelings, emotional states in larval zebrafish today. And I've picked this topic deliberately for today's talk because I think it sort of gives a very broad overview over what I personally believe it means to be a neuroscientist, which goes beyond, I think, what neurons do or what computations do, but it also extends to what defines ourselves as people, as beings, as humans, is this idea of thinking, of having thoughts, of having emotional states. And I think that's something that's still quite elusive to reductionist, mechanistic, hardcore, hard-ass neuroscientists, right? This idea of the things that psychologists find really appealing, namely feelings, emotional states. That's something that's sort of hard to grasp as a neuroscientist. And I think many, especially if you work with higher mammals like monkeys or rats or especially humans, we often fall into a trap that I sort of call closet dualism, where we think that there is something deep and mysterious about our brains that can never be explained based on the mechanistic basis of neuroscience. And the hope here is to try to connect these two things, to connect the mechanisms and computations that individual neurons do with sort of more higher cognitive internal states. So I think the power of the larval zebrafish here is not so much that it is small, translucent, readily accessible, and you can image every neuron in the brain while the animal is behaving. And just for the record, I don't think that's bullshit. I think what I mean is this whole brain imaging at single cell resolution. It's not bullshit, but I think it's much less useful than people give it credit to. I think it can be distracting at worst, and just relatively certainly insufficient to understand brain function at best, right? It might be a useful tool, but only if it's wielded correctly, right? I think doing brain-wide imaging and neural recordings without having very specific ideas and hypotheses, I think can be very dangerous and distracting. But anyway, so what I want to talk here is exactly, so the advantage of the larval zebrafish is, I think that akin to many insects, we are not tempted to anthropomorphize the fish because there are only 100,000 neurons in its brain. And so it's really difficult to take it seriously as a cognitive, holistic being with intense emotions because it's just so small and reduced. So everything you talk about in a larval zebrafish, it behooves you to talk about it in the context of a mechanistic reductionist and nervous system that and therefore I think it's a challenge to talk about feelings in this case and emotional states. And that's, I mean, the title is chosen deliberately in a somewhat provocative way. I should give credit to the people who actually did the work at the beginning. This is Caroline Wee, who did her PhD with me and was really spearheading most of all the work I'm going to talk about. But she had tremendous help and assistance with my friend and colleague, Sam Koens, who's also a professor at Harvard, and his co-scientist, Erin Song, who together with Sandy Wong added a lot of the initial ideas and experiments. So this is the credit. I also should say that Caroline really likes cats, so there will be a kind of a cat theme throughout of my talk, just so you're prepared. So first, this is my first cat slide that illustrates the power of internal states. So it just tells you that a lot of stuff can go on in the brain of an animal, in this case, the brain of these dogs, even in the absence of any outward behavior. Now, dogs are taught to sit still, but you can tell just by looking at these dogs that they are thinking a lot right now about this cat and what they'd like to do to that cat. So there's a lot of emotional states going on here, internal states that are not directly readable by outward behavior. There's probably also a lot of stuff going on in the mind of this cat here, right? That is, but I'll leave this up to you. I think one of the clusters we can sort of roughly classify internal states are the 4 Fs, yes, which is feeding, fighting, fleeing, and mating. Yes, and the these are sort of adaptive internal motivational states that I think we have in common with probably all vertebrates, yes, certainly all mammals. And it reduces the dimensionality of internal states to very few primary dimensions, which I think makes sense to talk about these 4 in particular. And it makes sense because all of them are obviously adaptive, and it allows us to link our scientific thoughts and experiments back to evolution. Right? Why have these brains evolved? To what purpose? And I think that sort of it sort of makes sense as a classification here. Yeah. So I mean, this gets me back to the problem of how we define operationally an internal state is. How can we tell whether an animal or an agent feels pain, feels fear, is stressed, or is lonely? Yes, because that is really the challenge here, is to convince you that a larval zebrafish feels anything, right? Like pain, hunger, or loneliness, or whether it's just a reflex machine that doesn't feel anything. I mean, this is sort of the deeper philosophical issue here that we have to grapple with when we talk about these things, right? You can, if you want, for later discussions, we can extend this even to machines and robots, right? When is it okay to talk about internal states and feelings in machines? So I've tried to come up with an operational definition of these things here, right? First of all, people told me I can't talk about loneliness in fish. I should call it an aversive internal state induced by social deprivation. That's allowed. So the right is allowed, the left is not allowed. And also whether fish feel pain or stress is also, I shouldn't use these words. They say, instead of pain, we should say an aversive internal state induced by noxious insults. And then for hunger, one could say, well, you shouldn't use hunger. I mean, this is me slightly tongue-in-cheek. We shouldn't call it hunger. We should call it an aversive internal state induced by caloric deprivation. And my point here is, to me, this is mostly a semantic debate. So this is a debate about words that we are allowed to use. And those are usually not very constructive and productive. So wherever you find yourself arguing about what words should mean, then I think usually it becomes less productive. So I will try now to use an operational definition for these internal states rather than psychological semantic meanings for humans. So here's my way of quantifying internal states. I say any agent, any animal has something that counts as an internal state if it changes depending on environmental context. So I think that's a necessary condition. An internal state needs to depend on the environment. So noxious heat for stress, food deprivation for hunger. So you should be able to introduce this or manipulate or modulate this state by an external manipulation. So, that's one condition. so it counts. The second one is ultimately there needs to be a behavioral readout, not like my dogs, which don't do anything, but in the end, throughout the lifetime of the animal, eventually there needs to be a behavioral readout. Like, for example, hungry, you're only really hungry if you actually eat more, right? As a consequence of your, or if you're afraid, you should shiver or tremble or run away, right? Or these things, right? Ultimately, there needs to be some form of behavioral manifestation. If not, it doesn't count, right? If it just happens internally, but there's never any manifestation, I think it doesn't count. And third, I think another necessary condition for it to be a card-carrying internal motivational state is that there needs to be some related neurochemical substrate, like a neuromodulator, a peptide that is causally related to this internal state. If you don't have that, then I think it's also probably not a mental state, but it could be a body state that that I think does not count. Yes. So this is more like a rough attempt to get a handle on what one is allowed to call an internal emotional state in any animal or entity. And I'm happy to take challenges and critique, but maybe at the Q&A session later. Whether you think this makes sense as a rough litmus test for whether something should count. But of course, on the other side, I would claim that if you can check off these 3 boxes, then it counts, right? Then it is an internal emotional state. And even if it doesn't feel like that for a human, doesn't really matter. All right, now let's use this and let's apply it to zebrafish, right? Loneliness in this particular case. How can you say that fish are lonely? Do fish have feelings? And I need to take a small detour here, and that is about not loneliness, but it is about appetite. Yes. Do fish feel hungry? Yes, larval zebrafish. Because the alternative hypothesis is that they're not hungry or satiated. They're just reflexive feeding machines that will just eat as long as there's food, and they only stop eating when they're running out of food. A little bit like teenage boys. Yes. So there's no sense of internal state. It's just a reflexive operation, and here is a technology we've used to show that this is not the case, that fish actually regulate their food intake based on the caloric deprivation state. And so I'll spend maybe 2 minutes to explain this, because we'll need this later on when we talk about loneliness as well. How can we quantify how much a fish eats, a larval zebrafish? And for that, what we do is we again take advantage of the small size and the perfect translucence of these animals. We label the food with a fluorescent dye. It's paramecia, it's small unicellular organisms that they hunt. And we label them with a fluorescent infrared dye. And then we simply count the fluorescence, quantal fluorescence increase in their stomach, right? We can do this in a 96 well plate. And here what you see is a hungry fish that's eating, and you can see that the stomach here is getting brighter and brighter, yes, while it's consuming these fluorescent paramecia. And so we can quantify this over time now, here over 2 hours. In black, we have a fed fish that we put into this chamber, and you see he doesn't eat as much, as rapidly, and his total meal size is smaller. If we fast the fish for 2 hours before we put them in, they eat with a higher speed and they eat more. And the 4-hour fasted fish eat a lot, and then they also eat much faster here. Yeah? Is that clear? So we can quantify how much fish eat, and again, this is going to become a relevant behavioral readout later on. The other thing we can sort of quantify is it's probably digestion and not pooping here, right? I mean, how quickly do the fluorescent food particles get removed from the animal? Now, the other aspect I mentioned is there needs to be an internal, this is back to loneliness, right? An internal neurochemical correlate of this, right? And in this particular case, as a neuromodulator in the larval zebrafish's brain, Caroline happened upon oxytocin. Yes. So oxytocin, most of you have probably heard of it. It is a well-known neuromodulator in mammals and humans. Yes, it's thought to be a cuddle hormone. It's related when we engage in social interactions. It's for child rearing, childbirth, nursing. It's active in these contexts. It's sort of supposed to be a social hormone. Yes. It is conserved, I think, across all vertebrates, and also larval zebrafish and zebrafish have oxytocin clusters in the hypothalamus, the same as mammals. Only there's much fewer here. Here there's the hypothalamus labeled with a specific line, the oxytocin neurons in the preoptic area up here, in the posterior tuberculum down here. There's about 20 oxytocinergic neurons on each hemisphere. And we can quantify activity in these oxytocin neurons, either by direct calcium imaging with two-photon microscopy, or we can use a sort of a cheap trick and doing phosphor staining. It's also sort of a proxy for activity, for cumulative activity with timescales of many minutes to hours. It's the poor man's calcium imaging. It has the tremendous advantage that you can do this in freely swimming animals without any constraints or without any invasive properties. Yes. So now this was one of the first experimental results of Caroline's work, is if we quantify oxytocin activity with this p-ERG method, we find that the activity in these neurons increases with stressful states. So the more we torture the poor larval zebrafish, the more active these oxytocin neurons become. Specifically, when we look at the ones in the preoptic area here of the telencephalon of the animal. And what you see is that activity in the preoptic area is increasing the more nasty the context, the treatment becomes. So electric shock gives you a lot of activity. Mustard oil, which is an abrasive toxic chemical that burns the fish, gives you higher activity. Noxious heat, if we heat them up, we see something. Dish taps, which are mildly annoying, give you some residual activity. An optomotor, which is a neutral stimulus, food, and even cocaine, which that's a different story, but larval zebrafish and adult zebrafish can react to cocaine in a way that seems like they like it and that they might get addicted to it. This does not modulate anything. So this argues that this preoptic area in the zebrafish scales with the negative quality of the context. So that's interesting, because oxytocin is supposed to be a sort of a positive signal, not a negative one. If I have time, I'll talk about this apparent controversy later. But maybe briefly, I think the resolution why we find that oxytocin codes for negative, for stress, and not for positive, like being in love and cuddling and making love, is because I think in most mammals, everything to do with childbearing, mating, nursing, and child rearing is not a positive state, but a very stressful state. Yes. Those of you with young children can relate to that. Those of you who have given birth can relate to that as well. Generally, I believe that everything with reproduction is probably primarily a stressful state, external state, not a relaxing state. And I think that's why I believe oxytocin, if anything, is more like an endorphin that allows you to deal with stressful situations. So that's our hypothesis, that oxytocin recognizes or encodes stressful contexts, and it will lead to escape behaviors. It will cause the animal to try to change its current context. In the larval zebrafish, just means to swim away. Yes. And to test this, what we did into the oxytocin neurons, and what we find is if we stimulate the oxytocin neurons, then fish will enhance their probability to escape. This is shown here. If we give a 100 millisecond pulse of blue light to oxytocin neurons, very little happens. But if it's a 5-second constant stimulus, the escape responses of these fish get upregulated. with a relatively slow time constant. So this is a modulator of escapes. It upregulates it. It does not induce escapes. This is in line with that oxytocin. It does not drive behavior directly, but it just changes the probability of releasing these. What we find here as well is that the response probability goes up with stimulus duration, and so does the response frequency, so bouts per second. Also, importantly, the latency for the stimulus, the mean latency seems to be around 2 seconds. So it's not a fast, it's a slow response. So that's the hypothesis. Oxytocin encodes stressful stimuli, and it relays that information to the hindbrain and spinal cord of the fish. And this is something we've tested here. Is oxytocin sufficient? Yes, the release of oxytocin, is it sufficient to elicit escape responses, even if the fish doesn't have a brain anymore, even if it doesn't have oxytocin neurons anymore? And so what we did here, we cut off the head of the fish. This is a headless animal now, where only the tail is still alive. And if we just add oxytocin to the water, yes, and allow it to diffuse into the brain here, into the hindbrain and the spinal cord, we see that after a while, these vigorous escape responses are elicited. So this tells us that oxytocin indeed has the ability to elicit these escape responses. And here's a movie of a headless animal. There should be a trigger warning here, that those of you with high sensitivity should not watch. This is a headless animal that's starting to first swim, does routine swims here. Oxytocin has been added, and you'll see that eventually it's starting now to vigorously escape, because the spinal cord here presumably is getting signals that via the oxytocin neurons to elicit these escapes. Yes, so what does all of that has to do with feeling lonely? Yes. This is another one of, this is Sandy Wong with Erin Song and Sam Kearns have initially done these experiments, and Caroline Hui has joined forces with them to look at this in more detail. And what we did is simply, same with the food deprivation, is ask, do fish, larval zebrafish, care if we socially deprive them? Yes. And the experiment is simple. We take 40 fish. They usually come in clutches. They are born in clutches of 200. So they're used to being in a crowd. If we isolate one of these fish, yes, and compare it to a group of fish that we keep in a group, what we find is that the isolated fish eats less. So this is one of the behavioral readouts that we find as a consequence of social isolation. And it is that they eat less food. And I've told you already how we can measure food intake. We add access labeled paramecia, and then we just observe how much they eat. And what we find is that a normal, this is how much a group-reared animal will eat, yes, after 2 hours of fasting. And here we see an isolated animal just eats less, and not just a little, actually less than half. Yeah. So this is a really strong effect. Social isolation leads to a decrease in appetite, if you want. They just eat less. So this is just, if we now play with the number of animals we put in this dish, we see that it really depends on the group size. So the more animals there are in the small dish, the more they eat, and the easier they get back to the original food intake. All right, so that's interesting. Yes, first of all, they care if they're isolated, and one of the ways it manifests is they eat less. Now, next we wanted to know, what are the signals they use? What are they cueing in on to know that they're lonely? Yes, are they looking, because they are very visual animals, do they look for conspecifics, or is it something else? And it turns out to be it's something else, because if you're trying to rescue the loss of appetite by giving them visual access to conspecifics, we put the isolated fish in the middle, and we have him or her surrounded by conspecifics, but with a glass wall in between. Then what we find is that the visual reintroduction of the conspecifics does not rescue the feeding phenotype. However, if we just take a few drops of what we call, well, of conditioned fish water, so a few drops of the liquid here and put them back in here, then we can rescue the feeding phenotype. Then they start eating again. That's why we've called this happy water. Yes, sir. So if we take happy water from here, put it back, the fish eats again, right? The appetite phenotype is rescued. So that's our hypothesis, therefore, that they smell the conspecifics. They need a certain odor in the water, or a certain chemical substance in the water that gives them the signal that they're not alone, and then they will recover their original behavior. All right? Now, is there any other behavior that is introduced by social isolation? And indeed there is. It is, they become, not only do they eat less, they also become a little bit more jumpy, a little bit more sensitive to noxious stimuli. And in order to test this quantitatively, we used Optovin, which binds to the TRPA1 channel. This is one of the canonical pain receptors in all vertebrates. We can activate this TRPA1 pain sensor with Optovin that we have caged, and we can uncage with light here. So this is just a very quantitative way of activating the pain pathway in these larval zebrafish. And what we find is that Optovin or TRPA1 activation will lead to escapes, as is not surprising and as is expected. But the lonely fish here, the isolated fish, will escape quicker and at lower thresholds than the ones that have been raised in a group. So being raised in a group also makes you not just more hungry and ready to eat, it also reduces your sensitivity and your threshold for pain. All right? Now, what about oxytocin? Right? So I've told you at the beginning that we think that oxytocin is an indicator for negativity of the context. So how bad is it for a fish to be lonely compared to these other noxious stimuli? And here again, you remember maybe the range of negative contexts we gave the fish, yes, which increases as such. This is in the preoptic area. This is in the posterior tuberculum where it's not so clear. And so this seems to be our benchmark here, where what we can use to calibrate. And you see that a socially isolated fishes has indeed increased oxytocin activity, which seems to sit roughly here between those two areas. So it's about as bad as noxious heat. As being lonely as a larval zebrafish is as bad as being warm. It's not as bad as getting electric shocks or getting noxious abrasive chemicals, but it is probably worse than being annoyed by dish taps here. Huh? I'm not saying this is to be taken in a truly quantitative way, but qualitatively we can sort of get now a metric of how bad it is, how bad a fish feels when you socially isolate him. Yeah, so it's a sort of a handle, a neurochemical, neuromodulatory handle. And if you look at individual oxytocinergic neurons and ask how much are they getting activated yes, when we apply these context changes, yes, you see that there is a lot of increase for shock, some for mustard oil, a little bit less for heat. Again, this is the the preoptic area up here and the posterior tuberculum down here. Taps a lot less, yes, sir. And groups of 3 also have very little oxytocin activity, but isolated fish and isolated with visual have more, yes, oxytonergic activity, and that can be suppressed again if we give the fish happy water, like water that's been conditioned with the chemicals that others have released into the tank. Now, if it's a chemical, then presumably, I mean, that's sort of the null hypothesis that they can smell it. So that was our next, or Caroline's next question. Can we find a olfactory bulb signature for these very specific odorances? And indeed, we can. This is now imaging experiment in the olfactory bulb of the fish, where we ask, are there neurons in the olfactory bulb that respond specifically to these different odorants. And here's one example of of neurons that respond specifically to conspecific odor, to happy water, that do others that specifically respond to adult water, adult conditioned water. Yes. One thing you need to know about this is adults are not good company for larval zebrafish because the parents will eat their young. So the adult is one of the predators of a larval zebrafish. So it is a predator queue. It is not a happy, my parents are in the vicinity queue. This particular and another neuronal cluster responds in particular only to food odors, right? So of course, that's also not surprising, but there are specific neurons in the olfactory bulb that are specific to conspecific odor. And if we look in the oxytocin cluster here, then again, not surprisingly, we find, I've shown this before, that, no, I haven't shown this before, right? Oxytocin activity increases with stressful signals. So adult odor will increase oxytocin activity. I haven't shown you that. That is interesting. What is also interesting that food odor seems to do the same, yes, which I think at least leads me to believe that if you suddenly present a hungry fish with food odor, it's not a happy stimulus, but it might be a stressful stimulus, which I think is not that unexpected, because if you're hungry and suddenly you're exposed with prey that needs to be hunted, this is a stressful situation because you don't know yet, right? The food is not accessible. It needs to be obtained. And again, there is a challenge. It's a challenge to the animal where it needs generally to be activated. Whereas if you give them happy water, yes, the odor of conspecifics, you get a reduction in oxytonergic activity, which should make the fish feel more relaxed and less impelled to do anything extraordinary. So we can quantify this effect of happy water on oxytocin neurons in more detail, and that's shown in this slide. And basically, the upshot is that it's not that easy. Oxytocin does not simply get repressed or suppressed by odor. Yeah, I mean, a lot of the neurons do, right? They are suppressed by happy water, but some of them are enhanced by odor. And for adult, it's the opposite, right? Most of them are actually enhanced, the activity by adult odor of the oxytocin neurons, but some of them are also suppressed. And for paramecia, for food, it seems to be more of a mixed signal, I say. So this leads us to believe that what oxytocin does, it is as a net effect, it does code for the negativity of the context, but it is has a higher dimensionality. So there's more variance amongst the oxytocin cluster, that some of these neurons might have very specific effects that go beyond a simple reporting of negative context. Yes. But generally, the model that we have, the hypothesis, holds true, and that is oxytocin is an indicator or a correlate of a negative aversive internal state. It can be induced by negative aversive stimulation, and it can be inhibited, this oxytocin, by the smell of conspecifics, which should remove the stressful state, because now they are in company. That is our model. If you followed along so far, you'll see that, or you'll notice that we now sort of require a pool of inhibitory interneurons that connect the smell of conspecifics, the happy water, to these oxytocin neurons. So whatever is inhibiting these oxytocin neurons needs to be represented by a pool of neurons that ideally are also driven, inhibitory neurons that are driven by happy water. And Caroline found them, at least a candidate, and these are GABAergic neurons that are located right between the olfactory bulb and the oxytocin cluster in the caudal hypothalamus. Yes, we have a fish line, a driver line that drives them. It's called Y321. And if we label them and do calcium imaging in these inhibitory neurons, what we find, there's indeed a subset of them which is specifically driven by, in this case, non-kin, actually kin-specific water, right? So whenever we give happy water, so water that's been conditioned with conspecifics, actually siblings, we get the strong response of these inhibitory neurons, right? That then would sit in a perfect position to relay this inhibition to the oxytocin cluster. So that is our current hypothesis, that that is the key player to relay this information. So, of course, if we characterize all of the response properties in this GABAergic cluster in the telencephalon, in the forebrain of the animal, we find, as you've seen here, the kin-responsive neurons. We also find adult responsive neurons, so that I think makes sense. That the presence of adults is also encoded in here. Yes, it's presumably, hopefully, suppressing something else. And then we have multi-responsive neurons as shown here. Right. So that's nice because that allows us now to formulate a sort of holistic picture of the larval zebrafish's brain that regulates its presence to conspecifics. So the idea is that oxytocin neurons are the hub, the central core that regulates stress, aversiveness, the need to change your context, to explore, to run away, to change the world. Yes, and if oxytocin is high, the animal moves away, changes, becomes active, engages. If oxytocin is low, it's more like the parasympathicus, probably also relayed through the vagus nerve, is the rest digest state of the animal, rather than the fight or flight that's elicited by the oxytocin neurons. The presence of conspecifics is being encoded by a social odor, yes, which we believe is relayed through these GABAergic neurons in the telencephalon that then remove the excitation from the oxytocin cluster and remove the state of stress in the animal. So, oxytocin activity then is relayed through all kinds of lower level neurons that lead to the appropriate behavioral actions. Now, the final set of experiments that Caroline did was loss of function and gain of function experiments with respect to these oxytocin neurons. So it is very specific predictions, especially about the feeding state of the animal. Because you remember, what we show is that when oxytocin is high, the animals, when the animal is stressed, when it's lonely, it will eat less. So oxytocin, that's what we believe, enhances the drive of the animal to explore, to run away, and reduces its drive to, when oxytocin is high, reduces its drive to feed. What we can do now is first do a loss of function. So what happens if we ablate the oxytocin neurons? Yeah? Ideally, the animal shouldn't be stressed anymore, and it should keep eating because there's no stress-induced self-starvation. Here is MTC is one way, is nitroreductase is one way of killing these cells permanently, the oxytocin neurons. So in a MTC control, that's the agonist that drives nitroreductase that kills the oxytocin cells. So this is the control where, if you remember, isolated fish eat less, group-reared fish eat a lot. If we ablate oxytocin neurons, then they all eat the same. So isolation does not lead to this social phenotype of eating less. You can rescue it. So. Another way of killing neurons is with Botox. You all know about this. So if we ablate the oxytocin neurons with botulinus toxin, we see that even the group eat a little bit less. Yes, probably Botox fucks them up somewhat. But again, the isolated ones are not that affected by the isolation anymore if they are lacking oxytocin neurons due to Botox ablation. And the maybe dirtiest way how you can reduce activity is with an antagonist, an oxytocin antagonist. And again, qualitatively, we get the same phenotype. If we block, in this case, oxytocin receptors, we also see that the feeding phenotype is rescued. Fish eat a lot again, even if they are reared in isolation. And the one gain of function experiment that we have is if you give an agonist of oxytocin, then what you see is that even if they're reared in a group, as said, they eat less, presumably because the oxytocinergic pathway is now activated by this agonist. So all of these loss of function and gain of function experiments go in the same direction, which is, of course, very reassuring. Right, so that's, I think, summarizes the main points I wanted to talk about. Again, I've summarized for you the reductionist mechanistic pathway that we have uncovered here that regulates feeding and loneliness, hunger and social deprivation. And what we can provide here is sort of a reductionist mechanistic way of studying these things that I think have all the necessary bits to allow us to integrate all of this into the field of psychology. And with that, I want to thank Caroline again, who did really most of the work. She has her own lab in Singapore now and is doing excellent. And I'll be happy to take questions. Florin, how am I doing?
Thank you, Florian. You're doing great. Or so I think. There are lots of questions. I also have a few questions myself. So perhaps I'll be selfish to begin with and ask a few questions independently.
You can speak a little bit louder, Florin. I think that.
Yes. I'm curious what you think of is oxytocin the whole spiel here? Do you think that the whole story about having an internal state and assessing the level of stress in the environment is given by these neurons? Because if you look at the effects, sure, you do see the changes in the right direction, but the effects are not striking as you'd expect.
Yeah, so of course not, right? I think this is, I was being maybe overly simplistic here in order to tell a simple story. There is a lot of neuromodulators that all in concert activate to in the context of stress. Cortisol is something that all of you know. Epinephrine, norepinephrine. It's a concert of many different neuronal ensembles that only together, I think, give you the full flavor. So yes, it's not the only. There are many others that play a role. I think oxytocin just is one of the pivotal key players, and I think it tells you that that by manipulating them in this way, you can get, in my view, really dramatic effects, which to us at least were also actually quite striking and very surprising in how clear the effects are, right? But I'm sure there is a lot of other neuromodulators that are involved.
Sounds good. I'll read a few questions from the audience and then.
Can I ask just maybe stop your screen share so we can see your face a bit better? If you want to show us anything in the slides, then sure, put it back.
Just share again.
I think it's nicer.
I'm not sure if that's a stressful stimulus or if it's increasing or decreasing the stress of the audience. So one question is oxytocin?
It would be interesting. What will happen with the oxytocin in the audience?
Yes, because if only could image them.
Sorry.
Yes. So the question is, if oxytocinergic activity conveys valence, can you use it in an associational learning paradigm? So basically expose the animal to a stimulus, activate oxytocinergic cells whenever the stimulus shows up, and analyze whether the previously neutral stimulus is then avoided.
That's excellent. That's an excellent question. I think it's, well, first of all, yeah, this would be wonderful. The reason why we have a hard time doing this is because our fish are really hard to train with any kind of associative or operant conditioning. So that's one of the dirty secrets of larval zebrafish, is they don't learn shit. So any associative paradigm that works well in any insect does not work in larval zebrafish. We've been struggling with that for 15 years now. I think God has cursed us for the blessing, on the other hand, of having an animal that's perfectly translucent and accessible, but they just don't learn. So otherwise, I think it might work. It, of course, would depend whether the learning process, the synapses that you're trying to change and modify, whether they sit downstream of oxytocin or upstream of oxytocin, right? It could be that oxytocin is really relatively late in the sequence and it just modulates the escape behavior, but it is not subject to what the correlates of the amygdala or the hippocampus in a larval zebrafish is. But my hunch is, if you could come up with any paradigm where fish will learn to associate an unconditioned with a conditioned stimulus, that activating oxytocin would count and would work as an unconditioned stimulus. It's a great idea.
Cool. Second question.
I can ask a second question if you want.
Yeah,
there's some of questions regarding the experimental preparation where you put multiple fish in the bowl and they're very happy. I think one of the questions was, is there a tipping point? Do they ever get too many? Do they ever feel stressed about being too many? And if there's a difference between having 3 fish together or 10 or 20 or 40?
Yes. So I think that was one of my slides where I show that 3 fish is a lot better than 2, is better than 1. 5 is a little bit better than 3, but not by that much anymore. And it seems to be it's about 5, 6, 7 fish, it starts to saturate. This is, of course, because our dishes are very small. So this has to be calibrated. So we can't really talk about numbers here. Actually, we should talk about density, right? About fish per milliliter and not numbers of fish. But since there's about 5 milliliters in it, I would say a fish per milliliter is sort of a relatively high volume, a high density that they are happy with. So that's the answer to that. And so yeah, I don't think if it gets too many, they don't seem to care all that much. And I mean, yeah, we haven't really tested it. Well, if it's too many, it probably also stresses them because they are irritated by mechanical interactions. So if they're forced to bump into each other a lot, I think they will display signs of being stressed and negatively affected. But that, I think, is independent of the olfactory cue. This is now a separate sensory input, like taps, yes, which I've shown you also, right? If you tap them constantly, you get an increase in oxytocin activity. And I think that's what will happen if you put too many in a dish.
So in your approach, Florian, you went to the fish to study feelings that maybe relate to a human, but is there any way to actually learn from the fish in terms of applying any of the insight to humans?
The question is, what do you want to learn, right? I mean, this is, I can talk a lot about this. So depends, yeah, it depends what you want to learn. If you want to help humans in terms of clinical problems, this is one of the laudable goals of neuroscience, is that we want to help and better the human condition by finding out what diseases plague humans, and we want to help cure those, right? I'm all about that. It's just that I'm personally not very good at it. So this is not my declared goal. My declared goal is try to get a mechanistic, true understanding of how circuits work, rather than trying to fix them. But again, so if your question is, can we use our fish to help the human condition in terms of disease, that's relatively straightforward. That's an easy sell, because in fish you can do high throughput screenings of drugs, of interferences, of schizophrenia, autism, any phenotype that manifests in mammals and humans, I think is likely reproducible in some form in a larval zebrafish. And then finding treatments is definitely easier in larval zebrafish than in humans. So that's relatively straightforward, right? If your question is, can we learn anything about how the human brain works? by studying a fish brain, then I think yes, absolutely, yes. And to me, it is the answer is it's actually almost impossible to understand how a human brain works because it's too complicated. I personally really struggle. The insights about fundamental principles usually come from simple organisms, right? Because they allow you to ask very simple questions and isolate hypotheses that you might have. This is very, very difficult to do in humans. So ultimately, I believe that oxytocin in humans works very similar than in fish. I think that even in humans, oxytocin activation might be primarily a response to a stressful stimulus. It's something we didn't know before. We have learned in the larval zebrafish. I think it applies, and it allows us now to think about the human condition in a slightly different way than we did before we have found out about these mechanisms in larval zebrafish.
I'm curious what you think about a different scale in time. So when you talk about feelings, at least from an introspective perspective, you do have spikes. They come and go and so on. And I would expect that as the environment would change suddenly, now they say that food is actually available, you would have a very intense, immediate change in feeling. And in the data that you show, there is indeed a change, but it is actually gradual. Is it
Yes.
is it the same? Is it different?
Yes. So I think that's the question. Whatever I call feelings, what is the filter, the time constant? So characteristic time constants of processes in the brain. And we know, all of you know, is that the characteristic time constant of neural signaling is on the order of milliseconds, right? It's the time constant of a spike. This is also the time constant of the fast processing in the brain and the time constant of immediate reactions, right? Like escape response to an electric shock in a larval zebrafish can happen within 5 milliseconds. Yep. I would call that not a feeling. Yes.
Okay.
I would call the sensation of immediate pain, yes, sir, and the withdrawal, I would say that's not a feeling, that's a reflex. Even in a human, if you burn your hand and you withdraw it, I would say in the first 100 milliseconds to the first 2 seconds even, you're not actually feeling pain. Yes, sir. You're just responding to a noxious mechanical stimulus, but it doesn't count as pain. Pain, I think, comes later when the low beta fibers, right, low conduction fibers become active. I think pain is a process, or feeling is a process that almost by definition has a low time constant. Yes, so I would say that the emotional part happens slowly with a delay and a low pass filtered version of the fast sensory process that comes first. That would be my sort of qualitative. Yeah.
But if you're hungry and if I show you food with the promise of giving it to you, you would change your internal state very fast. You'd be looking forward to it, and probably you'd be also changing your state of mind.
Well, that's the question. Happy. I mean, how long will it take my internal state to follow? Yes, at this pleasant surprise, is this within 10 or 20 milliseconds, or does it take a couple of seconds? And I would say it's the latter, especially if it's unexpected. If you suddenly get exposed to an unexpected stimulus, be it positive or negative, it usually takes a while for your brain to process this and realize and believe it to be true. Yeah? If you want to go all the way to human introspection, right? So I think that the emotional quality of our internal states usually has these characteristic lag times. But I mean, again, now we are truly introspecting, right? Which is really not allowed in.
Not allowed.
But it is allowed, but.
There's a question from the audience. Regarding the headless preparation, how much of the brain was removed? Also, are the oxytocin receptors elsewhere in the spinal cord that could be driving the behavior more directly?
Yeah, so everything above the hindbrain. The only thing we've left was the hindbrain. And we know that oxytocin receptors are almost everywhere. So they are in the midbrain, in the forebrain, and they're certainly in the spinal cord. There are oxytocin receptors in the spinal cord, and we believe that they are the ones that are actually responsible for relaying this activity.
One second, more questions. Maybe Adriana can ask the next one.
Yes. So there is one question that asks about the paradigm where you put a fish in a group and you see that they eat more. And can you actually correlate this with them being happy, or could this be just higher competition? So they want to eat more. There's less food to share between each other.
Yeah, so this is the, usually people who ask this question are siblings. Yes, sir. They're also usually male and they have brothers, and they associate being in a group when there's food available, not as something that's good, but that's bad, because you have to make sure. So I'm agnostic about this. The only thing we know, right, whether they are truly happy, right, independent of oxytocin activity, are they truly happy, or are they actually stressed out because there are brothers present who might eat their food? Yes, so that I don't know, but I think all the neuromodulatory activity that we can record, that's the whole point of finding a quantitative metric for internal state, right? That indicates to us that they are actually not stressed. At least they're not stressed, right? They are, the internal stress level measured by oxytocin is reduced, and they eat more. There was one other argument one can bring forth is, so are they eating more because they are worried about competition from the brothers? Well, I don't think that fish have a theory of mind like that, right? Why evolution has established these neural circuits, could very well be that there is competition, right? That fish just feel more like eating when there's others present, right? There's a drive to eat that's higher, and that's motivated by conspecifics.
And obeys both rules.
But I think,
competition rule exactly. And both happy water rule.
Water rule, yes, sir.
Okay. I had a curiosity myself. I wanted to see, you mentioned the comparison between how happy fish are to be together compared to how happy they are to be induced pain in them. And I wanted to see whether you tried the rescue experiment on the pain and noxious stimuli. So can you actually use oxytocin to rescue the noxious, the feeling of noxious pain? Can you inoculate pain by stimulating oxytocin?
You can. Well, that we don't know. We cannot. So what we can do, if the oxytocin, I haven't shown these data, if the oxytocin neurons are ablated, the threshold to noxious pain stimulus also is reduced. So they do not respond as well to noxious pain. The thresholds are much lower. I don't think they're inoculated to the pain pathway, right? So that's still active. The TRPA1 channels are active. The primary sensory ganglia are still active. So pain is still being recorded. It's still being processed.
Yes, but your metric was behavioral. It was the eating.
Exactly. So the eating is, and in this case, the escapes are reduced. So what we believe is that in order for fish to escape in response to noxious stimuli, oxytocin needs to be high. Otherwise, this whole escape pathway is just not sufficiently facilitated or sensitized.
Okay.
One other question. What is the difference between driving a certain behavior and increasing the probability that it appears in regards to oxytocin and escape behavior?
Yeah. I think that's sort of more straightforward to answer. I think driving the behavior is if I have a sensory motor reflex arc. Yes, like the knee-jerk response, or a blinking reflex to a looming stimulus, or a pupillary, or if you withdraw your hand from a hot plate. Yes, sir. These are all sensory motor reflexes. Yes, sir. And that's what I would say driving a behavior. If you anywhere within the reflex arc engage, either by driving the relevant sensory receptor, or by driving any of these neurons that sit in between these feed-forward reflex arcs. That's what I would call driving a behavior. Modulating the behavior, I would say, is if you have a modulatory peptide or a neuromodulator that can either facilitate or deprive this reflex arc. So modulation means that if I increase the excitability of one of these neurons and thereby make it respond more quickly. So that would be modulating it rather than driving it.
Another question. On the context of competition for food, one could expect that conspecific odor could actually increase the expression or the release of oxytocin. Any thoughts or observations about that?
Yes, yes. So I think there was the same question as before. This is about the competitive brothers, right? That eat my food. So I think the answer in this case is evolutionarily, because I think that the primary problem of a larval zebrafish is not scarcity of food, but getting eaten by a predator or actually being swept out of the nest. So I think the primary incentive for a larval zebrafish is not to find food, because food for a larval zebrafish is either abundant and then they don't have to worry about it, or it is absent and then there's really nothing they can do. Right? The only thing they can do is actually explore and swim out. So I think for food, it doesn't make a lot of sense for them to modulate their behavior. If they get lonely, if they lose their group, then they're in trouble. So this should trump the necessity for them to find food, also from an evolutionary perspective. So a fish is in trouble if it loses its conspecifics, or if it gets drifted out of the nest. It needs to get back to it. It needs to find its brothers again, or sisters. Whereas if there's not enough food, if food is sparse, it doesn't really help if you eat faster than your brothers, right? I think that's it's just a particular niche of the larval zebrafish. There might be other animals where this is very, very different, right? Where having too many conspecifics or brothers in the vicinity is actually stress and not relaxing.
We have another interesting question. Somebody asked whether you have ever tried unhappy water. So water where fish have been stressed.
Yes.
Does that work as well?
Yes. Oh, yes, that's a very good question. So the simplest form of unhappy water is the water conditioned by the parents, adult fish, right? This is also something that fish exude into the water, it's only adults, and that is definitely unhappy water. That stresses the fish out, it drives oxytocin up, it reduces their feeding, it changes the threshold. There is another much more famous unhappy water, which is called Schreckstoff, which was, I think, found by Frisch in the first place. No, who was it? No, it was... anyway, it's a substance that a lot of fish, release into the water when they get injured. And that is, for many, many teleost fish, is the strongest aversive cue that we know about. It really makes them freak out. The larvae might not have the receptor yet. So this is something that comes online in zebrafish at about 3 weeks, not before. Or at least it's much harder to get a robust behavioral response out of Schreckstoff application in larvae. It's something we haven't really tried yet. It's also really, really complicated because nobody has yet really found the pathway of Schreckstoff. What substance, what is it, and how does it work? But unhappy water, that's the short story. Unhappy water certainly exists, right, in many, many different forms.
I think one curiosity that the viewer was looking for is whether you transfer the water of a lonely fish. Does that make other fish unhappy as well?
No.
So a lonely fish is just as good as a happy fish.
Yes. It's not, there's nothing to do with the internal state of this animal. It's really just the stink of your siblings that they like, right? It doesn't really matter what state they're in. It might be fish pee or something like that. So we haven't isolated the substance that they release, but it is just the generic smell of conspecifics that they cue in on. It doesn't matter what state they are in. Yes.
Okay. And another specific question regarding the Y321 driver line. One viewer is asking whether that he knows that it's been associated with the regulation of social orienting, and whether you think this is mediated through oxytocin as well, or whether it's something else.
Right. So social orienting is the idea that fish will quickly, this is, I think, more like a sensory reflex, is that they will orient towards, at least larvae, and also adults, will orient towards other fish, or will orient towards higher density if there's groups. Yes, sir. I think that orienting response is primarily a sensory motor reflex, which is not directly caused by oxytocin. Whether it is modulated by oxytocin, I don't know. That's a good experiment, because in either way it would be interesting. One could predict that if you stress fish, that they're more likely to orient towards conspecifics, that they have a higher need to seek the presence of conspecifics. It could also be that it is unrelated. So I don't know.
Okay, thank you. Florin?
There's a technical question. How do you know that a change in calcium in these neurons equates release of oxytocin?
Oh, we have no idea. Yes, we are completely agnostic. We're just hoping that this general rule that an increase in calcium sort of tends to correlate mildly with an increase in firing rate. Yes. Mm-hmm. It's not a perfect predictor, as we all know, but it's not a bad proxy. Yes. So usually increase in calcium tends to correlate with higher firing rates. That's one thing. And the other thing we are sort of relying on here is that an increase in firing rate relates to an increase in release probability of vesicles. Yes. Which is also sort of the party line. I know it's not necessarily strictly true. So we are relying on these two assumptions here, which at least are not crazy, right?
They are not, definitely. Is there a difference in oxytocin release between fish seeing other fish when they are in the same tank, or when they are actually just watching them on a screen? So
yeah. So I think we think that in this particular instance, visual cues play a very minor role. So that it shouldn't matter, right? They need to smell them. Seeing them doesn't really do much. So, but I think the question was another one: can you fool a larval zebrafish that conspecifics are present visually? Yes, whatever that means. With virtual reality cues, and yes, we can. So these reorienting responses that were the subject of a question previously, they can also be elicited in virtual reality. That is something we can do. Yes.
Okay. So Florian, thank you very much. I think we're about to wrap this up as time goes by, and we've promised the viewers a specific time frame. But it was really interesting, and honestly, we cannot stop. It feels like we should go on forever. So thank you very much. And I would just like to say to everybody that we have another seminar ready for you on the 22nd of April. So this is in 2 weeks, also on a Thursday at the same time. So depending on your time zone, you can check. It will be Danko Nikolic talking about AI and how it accurately represents or does not represent the brain. So stay tuned, and we will send you some updates. If you registered on our newsletter, you will receive an automatic email. Otherwise, just follow us on the channels that you've followed us for now. So, Florin, if you would like to say anything at the end.
I would like to say thank you. Thanks, Florian. Very great, very good discussion. And I can only hope that the discussion will continue on many occasions by the Pike Lake.
Yeah, hopefully. And again, let me say thank you to all of you. This was a genuine pleasure and a privilege to speak here. And if there are more questions, I briefly looked into the chat. If there's more questions in the audience for me, just find my email address. It shouldn't be that hard, and email me if you have more questions or interest. But and again, thanks to Florin, Adriana, Raul, and
didn't do anything. All Adriana and Raul. Very good. Great. Now, be good. Enjoy.
Bye.
Stay safe.
So I'm leaving now, yes?
If you want to, you could also stay here and be lonely by yourself in the room.
You're only virtually leaving, Florian.
Okay, good.
I'm never leaving.